Okay, good afternoon, everyone. I hope that you can all see and hear me. We're going to carry on today with our discussion about DNA and DNA function. And of course, the most important thing that we need to discuss about DNA function, having looked at its structure and having seen how the cell replicates its DNA every time the cell needs to divide, we've got to ask the question, what is the DNA actually doing? Well, the DNA contains the recipes for all of the functioning of the cell. In that, the DNA contains the recipe for all of the proteins of the cell. And this running of and structuring of the cell is largely based upon the functioning of proteins, as we have already seen. But most of the enzymes that we've talked about have been protein. Most of the structure that we've talked about has been protein. Most of the functions that we've talked about has been tied to protein. The recipe for those proteins is contained in the DNA in the form of genes. For our purposes, we can assume that every gene has a resulting protein that can be produced from it. That's not, I must just mention, that's not strictly true, but for our purposes, by and large, we can say one gene, one protein. There are a couple of exceptions, but we learned early on in this class, and we've repeated it several times, that this is the central dogma of living things. All living things operate using this principle. The DNA is transcribed to messenger RNA, and that messenger RNA is then translated. It's translated to protein by these special units called the ribosomes. And that's that is going to be our topic of discussion today. There are two distinct processes that we need to talk about. The first is this, it is transcription. The second is this, it is translation. And they are completely different processes, but intimately linked because they end up producing the proteins that the gene has the recipe for. So the first thing that we need to talk about, we've talked extensively about the structure of DNA. We've got a good idea now how that nucleic acid is built. We now need to talk about this other molecule, which is RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. In many ways, they are extremely similar to one another, but there are some distinct differences. The first, these are structural differences, okay? First of all, RNA is fundamentally a single-stranded molecule. Where DNA was a double molecule, two molecules running anti-parallel to one another, united by hydrogen bonding across their bases. RNA is not. RNA is fundamentally a single-stranded molecule. I say fundamentally because you're going to see in a while that there is a complication to that. The second thing is that the sugar is different. Ribonucleic acid, it has a ribose sugar. DNA is deoxyribonucleic acid. It has a deoxyribose sugar. And the difference lies at that second prime carbon. That is the second carbon in the ring. In ribose sugars, there is an OH group, a hydroxyl group there, where in deoxyribose, that oxygen is taken out. That's why it's deoxy. So in DNA, at the sugar, there's just a hydrogen at the 2' carbon. The third major structural difference between them is that RNA uses a different base. Instead of thymine, which is used in DNA, RNA uses uracil, U. So wherever there would be a T in RNA, instead there is a U. It's very simple. So, and we'll see the difference in sequences in a while. So those are the three structural differences. But there is another major difference between DNA and RNA, and that is, if for our purposes, and for most of the purposes of the cell, there is only one kind of DNA. So DNA is DNA is DNA. But there are many kinds of RNA, and they perform many different functions. It's a fascinating molecule. We won't discuss it in any great detail, but these are the three major types of RNA that we are going to encounter in our discussion. And the first, of course, we've already heard. Messenger RNA is transcribed from the DNA. That is, it is a copy of the gene. It's complementary to the gene. And exactly what that means I'll show you in a while. But the messenger RNA is matched to the bases that are contained in the gene, in the template of the gene, of the DNA strand. So we'll see that in a while. So the reason for this is that there's no cellular mechanism for directly producing protein from the DNA. The cytoplasm, the ribosomes don't talk DNA. They talk RNA. And they can talk to RNA, but they can't talk directly to DNA. So a copy has to be made. A transcription of the gene has to be made, which is then exported so that it can be interpreted by the cell and protein produced. So that's messenger RNA. The second kind that we're going to hear about is transfer RNA. Transfer RNA is a very small molecule, but it's a very bizarre molecule because it's a mixture. There are many kinds of transfer RNAs because transfer RNAs actually transport amino acids. Amino acids become a part of the transfer RNA. They are bound to the transfer RNA as part of the molecule. So the transfer RNA is fundamentally, it's an RNA, but it has bound to it, it has a specific amino acid. In all of most living things, we only use 20 amino acids. So there are at least 20 kinds of transfer RNA, tRNA. In fact, there are more, as we'll see in a while. There are many kinds of transfer RNA, each carrying a specific amino acid. One kind of RNA, transfer RNA, can only carry that one amino acid. It's very, very specific. The third kind of RNA we are going to talk about is ribosomal RNA. And this is RNA, which is actually part of the ribosome. It has two roles. First of all, it is structural. It actually builds the body of the ribosome together with some proteins, ribosomal proteins. But there are several kinds of ribosomal RNA that go together to form these little bodies called the ribosomes. But that's only the one function. The other function is that this ribosomal RNA has enzymatic ability, and it is actually responsible for the process of translation. So here is RNA, which functions as an enzyme. We say that it is a ribozyme. So it has ribozymal activity. And I've said to you several times, when we talk about enzymes, most of the enzymes in the cell are protein. 99% of them, of enzyme activity, is by proteins. But the exception is ribosomal RNA and other RNAs which have enzymatic ability. These are only three, by the way. These are only three kinds of RNA that we're going to talk about. There are many others. RNA plays some very, very important roles in the cell. Okay, so those are the three main kinds. Here's how they interact. Again, it's our central dogma. So here is our length of DNA. And this length of DNA contains all the information for producing a protein. And that we refer to as the gene. And we will assume one gene, one protein. So locked into this DNA molecule here is all of this information. And the information is actually contained in the sequence of bases. You'll realize that even though there are only 4 bases used, 4 nucleotides rather, that are used, each with their different base, we can put those 4 together into an infinite number of orders. And the order of the base specifies something. The recipe for the protein is locked into that order. So the cell now needs to interpret this. So what has to happen is the cell has to go to the gene and it has to transcribe it. And it transcribes it into messenger RNA, a messenger RNA molecule. Now, before we move on, I will repeat this a couple of times because it's really something that you do need to know. When we look at a gene, we realize, of course, that there is that the DNA molecule is double. But the genetic information is only contained on one of those strands. And that we will strictly refer to as being the template strand. I may also sometimes call it the coding strand. Now, I just warn you that if you go looking at other textbooks, this nomenclature gets very confused sometimes. So we are going to stick strictly to talking about the template and the non-template strand, or the coding and non-coding strand. If I say template or coding strand, I mean that strand that contains the genetic information, which is transcribed. Okay, so the other thing I'll tell you, for any gene, just the one strand will be template strand, okay? But that may differ for different genes. You can go wander off to another gene somewhere remote on the chromosome, and you may find that it's the other strand, which is the template strand. But it will be one for a particular gene. It will be one strand only that contains the genetic information that serves as the template for the messenger RNA. So this process here of transcription is one that we will discuss in some detail. Okay. Once the messenger RNA is produced, it enters the cytoplasm. Now, it depends whether we're talking about prokaryotes or eukaryotes. Our interest is mostly with with prokaryotes, sorry. So as the messenger RNA is produced, it is liberated straight away into the cytoplasm. In actual fact, as transcription occurs, it starts to peel off as the messenger RNA strand grows. It enters the cytoplasm. And as soon as the messenger RNA appears in the cytoplasm, it will be latched onto by the ribosomes. And the ribosomes will begin to translate. And what they do is they produce a simple polypeptide. The polypeptide is the protein result, is the result of binding together amino acids, the monomers for protein. We bind them together in an order which is determined by the messenger RNA. And that order of the messenger RNA basis is in turn determined by the order of the bases on the gene. So we go DNA, messenger RNA, protein. That is the central dogma. All living things use that methodology. So we want now just to talk a little bit about this process of transcription, because this is the first critical step in the whole process, in the whole, excuse me, in the whole central dogma. And we need to think about this a little bit carefully, because one thing you need to remember is that genes are only read off as they are needed. Now, some genes, some products of the gene may be needed all the time. Those we call, and the genes that are read off like that continuously because their product is needed continuously, we call the housekeeping genes. And those are things, for instance, essential enzymes that are used the whole time. Enzymes, respiratory enzymes, for example, may be read off the whole time. Other genes are only read off on demand. They're only transcribed intermittently. So there have to be ways of switching genes on and switching them off. Genes are kept under strict control by the cell, because otherwise the cell could be producing inappropriate proteins, protein product, and also just wasting energy. And when genes that ought to be switched off are kept switched on, that produces a disease state. This is characteristic of sometimes of infections that cause certain genes to be switched on and just read and read and read and read a particular product. But they are also characteristic of conditions like cancers, for example. Genes begin to operate inappropriately. Normally, the cell keeps very, very careful control over which genes are transcribed and when they are transcribed. That control mechanism is very complicated, but if we have a look at just at a gene, a length of DNA which is going to be transcribed, we'll see that first of all, the enzyme complex that produces the messenger RNA copy that does the transcription is actually an RNA polymerase. Right? As soon as you see that polymerase, you know that something is being polymerized. In this case, it's RNA nucleic acid. We saw before DNA polymerase. Now we're talking about an RNA polymerase. So all of this, the transcription, the production of the messenger RNA is going to be done by this RNA polymerase. And it's here at its simple. This is actually a complicated story, but I'll just make it as absolutely as simple as possible. It's here that the cell exerts tremendous amount of control over a gene, because what happens is upstream of the gene itself, before the gene starts, there is an area of sequence which is called the promoter. And this is an area where the RNA polymerase actually binds. And normally, this binding of the RNA polymerase is very carefully controlled. And there are a couple of different factors that have to be operating in order for this RNA polymerase to bind tightly to the DNA. And it binds at this promoter. There's a little bit of sequence there, which is recognized by the cell. And under the correct conditions, when the gene needs to be switched on, the RNA polymerase binds tightly here. And it then moves down. It doesn't start transcribing yet. It binds upstream and it moves down slightly. And then, only then, it is given an instruction to begin transcription, to begin the process of transcription. And this is, so now let's just have a look at this diagram here. Hold on, I've got to move the video thing out of the way. So have a look here. Here's our DNA strand. We're looking at the start of the gene here. Here is the promoter sequence there, where the RNA polymerase actually originally bound. the RNA polymerase moves down, and where at that point where it has to start the transcription process, it splits the DNA strands apart from one another. Remember, it's only going to produce messenger RNA from one of those strands, and that is the template strand here. So it is now, this RNA polymerase is moving in this direction. It's moving in that direction there, that way. It's opened these up like this. And what you immediately realize is when it does that, these bases are opened. They are single. Now, if those bases are single like that, they can be matched by free nucleotides in the cell. And that's exactly what the RNA polymerase does. It catalyzes a reaction where matching RNA bases, not DNA bases, but RNA bases are brought in. These bases differ from the DNA. First of all, look here. Here is on the DNA, on the template strand, here is adenine. What is it matching to in the RNA, in the messenger RNA? It's matching to uracil, not to thymine, but to uracil. In the DNA, there's its match across the other side. It's been split apart from thymine there, but in transcription, uracil is brought in. And these here, the backbone here of the messenger RNA is formed from, in the same way as it was in DNA, it's formed from a bond between phosphate groups and the ribose sugar, not a deoxyribose, but a ribose sugar. So there's a ribose sugar phosphate backbone to the RNA, and it uses uracil, not thymine. If we see A in the DNA, we'll see a U in the transcript. Now, this here, this process here actually only opens up relatively few of the DNA bases at a time. And the process of transcription is slower than the process of replication. Replication takes place extremely quickly, but the transcription takes place relatively slowly. And it produces a strand of messenger RNA. As soon as the polymerase leaves an area, the DNA simply winds up behind it. It is enzymatically controlled so that it is done absolutely carefully and correctly, but the messenger RNA strand is freed like this. This carries on going down, and in actual fact, if need be, several RNA polymerases can run down a gene at the same time, producing several strands of messenger RNA. So this is our strand of messenger RNA here. Eventually, the RNA polymerase will reach an area which is called the terminator. And that is where the gene ends. And when the RNA polymerase reaches the terminator, it falls off the DNA transcript, and the whole DNA strand winds up, and the complete messenger RNA strand is liberated. So let's just do it all again once more. The RNA polymerase, which is going to be responsible for transcription, initially binds at the promoter. That is where that process is how the cell exerts really careful control over which genes are being transcribed. It moves down to the start of the transcription area, and it opens up a small area of the DNA, splits the helix apart. And on the exposed template strand, it lays down an RNA strand. This RNA strand is complementary to the DNA bases. Look here, A, U, T to A, C to G, G to C, okay, etc. So this is complementary, but this is a short area of where this kind of hybrid strand exists, because the messenger RNA comes free from the DNA strand, and the DNA strand winds back up behind the RNA polymerase. It moves through the whole gene until it reaches this area called the terminator. And when the terminator is reached, the RNA polymerase is liberated, and so is the messenger RNA strand, the completed messenger RNA strand. So it's a relatively simple process. It has some similarities to DNA replication, excepting that when we open up the DNA strand here, we only read off one strand, not both, and we produce a messenger RNA strand, not another DNA strand. So that is the whole process of transcription. And now I need to just have a look at the actual structure of this messenger RNA strand. The first thing to say about it is that it actually contains a recipe, which is the transcription of the DNA recipe. But the words of the recipe are in units of 3. In messenger RNA, a 3-base sequence is called a codon, and codons are specific for something. Most of them are specific for an amino acid. Some of them are specific instructions to the cell, especially to the ribosome, as we'll see in a minute. So in messenger RNA, the 3-base unit is called a codon. That codon is produced, of course, from DNA. And in the DNA, if this, let's just say that this is our codon UAG in the messenger RNA, that is produced from a triplet in the DNA. The DNA triplet ATC produces the codon UAG. And it's really important. I'm afraid to introduce this to you, but you need to keep really careful note of the priming of this. We need to follow where the primes are. So let's just have a look at the production of the messenger RNA. Where are we adding bases? We're adding them here. This is a nucleic acid. The only place we can add other bases is at the 3' end there. That is the 3' end of the messenger RNA strand. So when I look at this codon here, UAG, there's the 3' end, it would be, and there's the 5' end. If I was to write this codon down, I would say 5' UAG. If I was to write down the triplet, the DNA triplet, I would write down 3' ATCY, because they are anti-parallel. 5' there must be matched by 3' there. So just we'll see this again and again. I'm just introducing it here so that you don't get confused when I start referring to this, because it's a good way. It's a way in which we are going to actually keep track of how different things match up to one another. Okay, so we've got 4 bases. I won't do the math, I'll just tell you. We've got 4 possible bases in our messenger RNA: cytosine, guanine, adenine, and uracil. If I've got 4 bases, but every word, every codon is made up of 3 bases, I do the math, I end up with 64 possibilities. There are 64 possible codons that can be. We only need 20. We only have 20 amino acids that we're going to use to make proteins. So why is there this? Why is there this disparity? We say that first of all, this is a degenerate code. That means any one amino acid can be coded for by several different codons, as we'll see in a minute. So we say it is a degenerate code. It has no pejorative association. It simply means that any one amino acid may have several codes for it. When we look, here is the actual, let me just, I always have to rearrange all of the little videos because it blocks things on my screen. So here is an actual, this is called the table of codons. Immediately in your brain, please, or if you have a written copy of this, then please write here in big letters, right there, 5'. 5'. Okay, because this table of codons is the genetic code. But this is a table of codons written in RNAs. It's written in RNA language. So when we have a look, you can immediately see, because it's U's. Look at all the U's here. When we look at the codons, this side here is the 5' side. That there is the 3' side. And I'll point out why I'm making a fuss about this in a second. So we got 64 possible codons. We've only got 20 amino acids. What we find when we look at this whole table is that 61 of these codons code for specific amino acids. And when with 61 amino acids, we're left with 3 that are not coding for an amino acid. What are they coding for? Have a look here and you'll see UAG, UAA, UGA. These code for the stop codon there. UGA, UAA, UAG. Those are called the stop codons. And that is like the full stop at the end of a sentence. During the process of translation, when we see one of those stop codons, translation is going to end. So there is another codon which has a dual function. And it is this one here. This is almost the only codon I'm going to expect you to remember. Okay. You will always be presented with a table of codons to work from. When I give you problems or whatever to solve, you will have a table of codons to work from. So, but this one you need to remember, AUG. That is called the start codon. And it is a particular AUG codon marks the beginning of translation for the ribosome. It marks the big start of the process. And the process ends at a stop codon. And in between, we can use all of the codons that are for the other amino acids. Now, here's why we say this is degenerate. I'm actually going to flip from here to this one here, to this table here. I know that this is not in, if you had printed out the PowerPoints before, this is not there, but I have posted this table to Canvas so that you can print a copy of it when you're studying. And whenever I promise you that whenever I set problems or anything, I will give you this table. And the reason is I don't expect you to memorize all of the abbreviations for the amino acids. Now look through here and you'll see that there are the 20 amino acids are all represented, but many of them are represented by several different codons. So what we have here, for example, here's phenylalanine there. And phenylalanine, PHE is its abbreviation. Phenylalanine is represented by the codon 5' UUU, 5' UUC. Either of those will result in phenylalanine being used. Leucine has lots, has all of these there. Here's isoleucine, those three. Methionine only has this one. the AUG, and so on and so on. So first of all, if you are presented with a problem and you are asked, what does the codon CAU code for? Well, what you can do is you can chicken pick through the whole thing. That's fine. There's no reason not to do it. CAU, 5' CAU, there it is there, 5' CAU. And that codon codes for histidine. There it is there. But this contained in the structuring of this table, there is another way to do it. So if I say the CAU, all that you do is come to this line of bases here, and you'll find the C. There it is there. Your answer is somewhere in this block here. It's somewhere in the C block. That's the first base, right? The second base, I said, is A. So there is the A column there. And here's where the C row and the A column intersect there. You can already see it. But here's the third one here. I said CAU, there it is there. See, so we go CAU, and the only place in the whole table where they intersect is there. And it tells you this is histidine. Okay, so there, it's on the face of it, a relatively simple code, very, very profound, however, because locked into this code are the recipes for all of the proteins in all of the living world. They all use this table of codons. It doesn't matter whether you're talking about a jellyfish or gorilla. All living things use this table of codons. This table of codons is a deciphering of the genetic code. But what I want you to remember most of all about when you look at the table of codons, remember this is a table of codons. Okay, it is table of messenger RNA. It is not the table of DNA triplets. Don't get confused. Otherwise, you won't be able to answer any of the problems that you get set. So 61 code for amino acids. The order of the codons is going to determine an order to the amino acids. And it's going to determine the order in which they are bonded to one another to form the polymer that is a polypeptide. Three of them, these three here, UAA, UAG, and UGA, are the stop codons. We need to say a word, though, about the use of this methionine as a start codon. And the first thing I need to point out to you is there is only one methionine, which will serve as a start codon. But methionine is a very common amino acid. It's used in all proteins, and it occurs very frequently. So we need to answer the question, how does the cell know which of those methionines is actually the start codon? And to do that, we need to proceed. Okay, there's also a little animation for you. I won't run it now. There's an animation for you. The process of reading off the messenger RNA and producing a protein from it is called translation. And the process of translation will begin at an AUG, at a particular AUG. And which one is the start codon is determined by a little sequence in the messenger RNA. Now, the messenger RNA is very long, and it's not solely, it does not only contain the information to create the polypeptide. There are, on either side of that information, there is other information in the messenger RNA. We won't discuss it in enormous detail, but there is a lot of information contained in the messenger RNA. And one of the pieces of information is a little short sequence, which is called the Shine-Delgado sequence. And the start codon is the very first AUG after the Shine-Delgado sequence. So that's how the cell knows. And we'll see in a minute exactly how the Shine-Delgado functions. So I also want to just quickly run you through maybe some simple exercises in how to consider the different codons and how they are related to, for example, to the DNA. So if we come back, I just want to come back here. And I want to ask you, if I have a sequence, which is AC, I'm just going to write it on myself, ACU and AAU in the messenger RNA. I've got 5' ACU and AAU. What two amino acids are going to be joined together by those two codons? And we go ACU, and here we are, here it's threonine. And then what did I say? AAU, and asparagine. So we would have threonine and asparagine would be joined together. That's fine, that's easy. But now let me ask you something. What are the DNA triplets that produce those? ACU and AAU. What DNA triplets produce those codons? I'll give you a minute. The answer is TGA, A to T, C to G, U to A. So it's TGA, and here T, T, A. Those are the triplets, the DNA triplets, and they would read 3' TTA, okay? 3' TGA to this 5' ACU, 5' AAU. You must get used to being able to shift back and forth. If I ask you, if I say to you, I've got a DNA triplet which reads this, what protein will eventually be, what amino acids will eventually be produced from that? What will the cell, how will the cell respond? What amino acids would it put in if I had these DNA triplets? I might even ask you, for example, if I have a non-coding strand of DNA that reads such and such and such and such, what is the mRNA that gets produced from the coding strand? We need to be able to shift. So what I will do, I will, sometime in the next day or so, I will give you a short problem set to take you through these kinds of things. Whenever you are doing those problem sets, always keep a careful eye on exactly which nucleic acid strand you're talking about. Are you talking about the non-coding strand? Are you talking about the coding strand of DNA or template strand of DNA? Are you talking about messenger RNA, or are you talking about some other kind of RNA? The cell has to make these decisions too. And so our next section that we need to talk about is how exactly does the cell now respond to the presence of messenger RNA? And how does it go into the next process whereby it takes the sequence of codons and actually translates it into a sequence of amino acids? And the answer lies in this amazing little molecule called transfer RNA. And transfer RNA, let me just come back here. Transfer RNA has is also a nucleic acid. It's also a single-stranded RNA, which means it has a sequence of bases. Now, that sequence of bases is really important. I'd say there are two very important parts to the transfer RNA molecule. And the one is the 3' end, because at the 3' end, there is an amino acid attached. And if we then go all the way through the molecule, I thought I had. Okay, I'll just use this as an example. Okay, this is a transfer RNA molecule here. So here's the 3' end there. And this has, in this case, this has an amino acid attached to it, and that is methionine. There it is there. If we go through this RNA molecule, the transfer RNA molecule, we reach this area here. This is the business end here of the transfer RNA molecule, because there is a short sequence here, UAC. It should ring a bell, because methionine, what is the codon for methionine? The codon for methionine was AUG. This matches it, A to U, U, A, C to G. This is called the anticodon. So this anticodon matches a codon here. It will actually bond to it by base pairing. It'll base pair U to the A to the U, C to the G. And this anticodon, a transfer RNA with that anticodon, can only carry methionine. Now, this methionine is bonded to the transfer RNA in the cytoplasm. As these transfer RNAs are created, or manufactured rather, with their specific anticodons, a specific amino acid is bonded to them. And the amino acid always matches the anticodon, and the anticodon matches the codon for that particular amino acid. So, for example, transfer RNA for phenylalanine has an anticodon which reads AAA. The anticodon is AAA. So what is the codon that it corresponds to? It is UUU. There it is there. The codon UUU will match to a transfer RNA with an anticodon AAA. Our table of codons is always the messenger RNA. Okay, but from there, we can tell. From this codon, we can tell what was the DNA triplet that it matched to, that produced it. What is the transfer RNA that it's going to interact with? What is the transfer RNA anticodon that is going to interact with? We know those. We can work that out almost instantaneously because we know the base pairing rules. A to U, U to A, and C to G, G or G to C. The simple base pairing rules still apply, no matter what species of nucleic acid we're talking about. Same, here's another one, is tRNA for serine, has the anticodon 5' UCA, binds to this codon, okay? So let's just talk about now about the process of translation. There are 3 stages to the process of translation. The first is that translation has to be initiated. And translation is initiated by the binding of ribosomal subunits to the messenger RNA strand. Once translation begins, the ribosomes will begin joining amino acids together. So then there is a long period of elongation. And when the ribosome runs through the entire messenger RNA strand and produces the polypeptide. And then there has to be an ending. a termination. And that termination is when the ribosome encounters a stop codon on the messenger RNA, where as soon as it sees a stop codon, the ribosome comes away from the messenger RNA. So there are a couple of steps to the process of initiation. And they're as follows. You'll see normally in the cell, in the absence of messenger RNA, there are 3 ribosomal subunits. The ribosome consists of 2 parts, a small subunit and a large subunit. And they're normally free from one another. As soon as messenger RNA appears in the cell, the small subunit actually binds to the Shine-Delgado sequence down here somewhere. It binds, and as soon as it's bonded, it moves, and it moves here. And to this area here, it finds the first AUG, the first codon for methionine after the Shine-Delgado. And it stops there, and it catalyzes the bonding of the large subunit. So this small subunit has some particular responsibilities, has to recognize the messenger RNA, bond at the Shine-Delgado, come down here, and make sure that it is correctly positioned over this AUG, because that determines the position of this large subunit. And to understand why that is important, we need to just take a look inside the large subunit. And we'll see that there are 3 different sites. This one here is called the A site. This one here is called the P site. And this is the exit site there. So that's A site there, the P site here, and the exit site there. Those are their names. There is actually, it's not shown here, but there's a little tunnel here in the large subunit for the emerging polypeptide to squeeze through. So this is the way it works. This small subunit positions itself here in exactly the right place, and the large subunit comes here so that it is sitting with the P site right over this AUG start codon. There are some rules to obey. The first rule is that there can never be an unmatched codon. Every codon that is exposed in either of these sites must be matched with something, must be matched to an anticodon, or something else will happen. So when we first put this large subunit here, this AUG is sitting there with no tRNA associated with it. As soon as that happens, the cell will bring in a tRNA, and the tRNA it brings in matches the codon. Its anticodon matches the codon that is exposed. So we now bring in this specific tRNA, and that is the tRNA which carries methionine. Now, the other thing that is not tolerated is an empty A site. As soon as there's an empty A site, the next tRNA will be brought in, and the tRNA that is brought in matches this exposed codon there, UUA. So the anticodon must read AAU. Let's just go back. Let's just remember this: UUA, 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 UUA. There it is there, and it's coding for leucine. Okay, so that is the tRNA coming in here. Here's the tRNA with its anticodon AAU matching UUA, and it's carrying leucine. There it is there. It's going to sit here And what is going to happen now, this is a critically important step. And unfortunately, our brains sort of get it twisted around sometimes. What we've got here, we've got two tRNAs sitting right next to one another. And we've somehow or other got to form a bond between their amino acids. And that is the next step. Because what happens now is that this methionine here bonds here. Don't make the mistake of thinking that this is going to bond there. It doesn't. This methionine bonds here to leucine, and it leaves this empty. Here's the next rule. The ribosome will not tolerate an uncharged transfer RNA. It will not tolerate the presence of an RNA, a transfer RNA, without its specific amino acid. This specific amino acid now leaves this transfer RNA. It goes here, leaves this uncharged. The response of the ribosome is to move. It's to move one codon down. And that relative movement down the messenger RNA is called translocation. So that we join our methionine there, we leave that transfer RNA empty, and the whole ribosome moves one codon in that direction. And it puts that empty tRNA here in this E site, the exit site, and it is kicked out. Okay, now what have we done? Well, we've moved down and we put an empty A site there. We moved the ribosome over a new codon, GGU. Well, that's not going to be tolerated. GGU, the anticodon matching that is CCA. You can go back if you want and look them up, and you'll see that it is for glycine. There, this tRNA carries glycine. Immediately that happens, this little dipeptide here, leucine, methionine, this moves and bonds there. The bond that it makes is a peptide bond, a specific bond which joins amino acids together and begins to form a polypeptide. Now remember, if I move this little dipeptide here and I bond it here to form a tripeptide, then I've left this empty. Our E site is empty because we kicked out this tRNA, but our ribosome won't tolerate an empty tRNA. What does it do? It translocates. It moves another 3 down. And the next one, the next codon here is going to be UUU. Here's the anti-tRNA ready for it with its anticodon AAA, and that is going to code for phenylalanine. Phenylalanine will be the next one. And in the meantime, the translocation will have put this empty amino acid here in the E site, and it will be kicked out. And so on, and so on, and so on, and so on. Step by step by step, by translocation, by translocation, by translocation. The ribosome takes the code in the codons. It brings in matching transfer RNAs in the order described by the messenger RNA, and it joins together specific amino acids, which all together form a polypeptide. That polypeptide begins to get expressed out of the ribosome, and eventually it just carries on and on and on and on and on. Eventually, the ribosome is going to reach a stop codon there. And when the stop codon is reached, a set of events occurs. There's no corresponding amino acid or anything, but there is a protein and a set of factors called the releasing factors, which come and bind here at the stop codon. This could be one of any of the 3 stop codons. They all work the same. Once that releasing factor occurs here, this polypeptide is released from the last transfer RNA, and the whole system falls apart. The 2 halves of the ribosome come apart. They go off to do their work all over again, grab onto another messenger RNA. This transfer RNA here, all of the transfer RNAs can be recycled. There is a cellular mechanism whereby these empty transfer RNAs are matched with their correct amino acid and are recharged, ready to start in another translation process. So there is a little movie for you to watch. I will tell you there are some differences between eukaryotes and prokaryotes. Our interest is mostly on prokaryotes. This little movie is about eukaryotes, but the principles are the same. I will later on also point out to you a couple of differences between the processes, between prokaryotes and eukaryotes. Remember that in the prokaryotes, this entire process is taking place in the cytoplasm because the DNA is inside the cytoplasm. The DNA is not contained in a separate organelle. That's a big difference because it means that this messenger RNA strand, when it's transcribed from the DNA, goes straight into the cytoplasm and can emit even while transcription is taking place. Ribosomes can grab onto the messenger RNA and they can begin reading it. And as a ribosome moves along the messenger RNA, creating the polypeptide, another ribosome can come behind it and start the processes of translation as well. So a single messenger RNA strand may be translated many times. Messenger RNA usually does not survive very long inside the cell. Minutes or hours would be the time span. It's soon degraded, and that is a control mechanism. We only produce the messenger RNA when the actual protein product is required. We don't want messenger RNA hanging around forever, producing unnecessary proteins. So when we look at prokaryote process, we've got a whole process, transcription and then translation, taking place inside the cytoplasm. This is an amazing photograph. This is an electron micrograph. And what is happening here is that this is the DNA strand. You see this very faint line there. That's a DNA strand. And this is a gene along here, and RNA polymerase, which you can't see, it hasn't been stained. RNA polymerases are running along, and they are producing messenger RNA strands. And then as the messenger RNA strand is produced, ribosomes, these little round bodies, are grabbing onto the messenger RNA, and they are producing protein, which you cannot see here. So this is an example. From one gene here, we can produce many strands of messenger RNA, and from one messenger RNA strand, we can produce many strands of polypeptide. So it's a very efficient process. The one thing I do want to point out to you, though, I just want to ask you, which is the oldest? You see here, these are each represents a strand of messenger RNA being read off the same template. Which is the oldest messenger RNA strand? This one here, the longest one there. And if I was to ask you for this longest strand here, which ribosome, each of these is a ribosome, which ribosome has been on there and produced the longest protein strand from there? And the answer is this one here. This messenger RNA down here is only just being created. As soon as it's created, it's grabbed onto by ribosomes, but they've only had time to produce a little bit of protein. These other ribosomes have run all the way down here. They've run in this direction down. They are running in this direction down the messenger RNA, producing their proteins rather, and this one's about to fall off. It's about to reach the stop signal, and it'll fall off. So this brilliant picture shows us a huge amount of information. And these in this kind of structure is typical of the prokaryotes, and it arises because these processes all take place inside the cytoplasm. And this is called a polysome, and it's specific for the prokaryotes. Cannot occur in the eukaryotes, because in the eukaryotes, the transcription takes place inside the nucleus, and the messenger RNA must be exported from the nucleus to the cytoplasm where it is translated. Okay, so that's not the only difference with the eukaryotes. In the prokaryotes, the messenger RNA emerges mature. It can immediately be utilized. The message is simple and straightforward, and the ribosomes can grab onto it and immediately start the process of translation. And in eukaryotes, however, there's a bit of a difference. The messenger RNA in eukaryotes actually has areas which are going to be translated, and then interspersed in between them, there are areas which are not translated. It's not nonsense. They actually contain, as it turns out, they contain a lot of information of different kinds, but there are only certain areas of the messenger RNA transcript in eukaryotes which are going to be translated into protein. Those areas are called the exons. And the intervening bits that are not translated are called the introns. And before the messenger RNA can be translated, those introns are edited out. They're cut out, and the exons are all joined together. And then there is also in the eukaryotes, the two ends of the messenger RNA strand have specific information added to them. And all of that takes place inside the nucleus. And once the messenger RNA is actually mature, we cut out the introns. Here, take these out, join everything together, and then this messenger RNA strand has tails added to it on either side. And part of the reason for adding those tails is to give the cell information about where the messenger RNA needs to go. And so it is then exported from the nucleus through the nuclear pores and into the cytoplasm for its own translation. Okay, so I'm going to stop there for today for a specific reason. I want to actually, this is the next slide we're going to do if you're reading ahead. I'm actually going to see if I can amplify this information a bit, because this is too abbreviated here for it to be really clear. So at your next class, we'll start here. And in the meantime, if you have any questions, please feel free to ask. No?
Professor, I have a question.
Go ahead.
Yeah, just to recap on the priming. So DNA, it starts with 5'3" priming, and then mRNA, it's 3'5".
Yes, don't forget that the DNA is a double molecule, and it's anti-parallel, right? So one strand 3'5", and then the other strand must be 5'3". because it's anti-parallel. The same rule applies when I start to, for instance, to make, let me get back here. I'll go all the way back to here. Okay, so the same rule is going to apply when I start to polymerize another nucleic acid that's complementary. So what I'll point out, if you ever get lost, always look at the strand where something is happening. Here, this is our RNA strand. We are adding bases there. The only place we can add bases is the 3' end, right? So that's the 3' end of this messenger RNA strand, and its 5' end is here. Okay, so it's running 3' to 5'. And this, if I was to look here, what is the priming here? This must be a 5' on the DNA. Okay, that must, its 5' end is here. Its 3' end is there, down there. Its 5' end is up there, because this is 3' to 5'. So this DNA strand, if we were to follow it all the way to its end, we'd end up at its 5' end on our right-hand side, because it has to match the 3' end here of the messenger RNA. But remember that messenger RNA is single-stranded. Does that clarify it for you?
Yeah, I got it. It's
okay.
Basically the 3' is where you're adding the bases, and then 5' is already where it started.
Correct. Yes.
And then it's opposite to the DNA.
Correct. As soon as you have complementary nucleic acids, whether it's 2 DNA strands making up the whole molecule, or if we're talking about like in transcription, where we've got a hybrid between DNA and RNA, the nucleic acid strands always match 3' to 5'. They're always anti-parallel.
Okay, got it. Thank you.
Mm-hmm. Anyone else? Okay, and remember, over the next couple of days, I'll set a short problem set for you. And that is just, it really illustrates the principles of the transcription, of replication, transcription, and then of translation as well. Okay.
Professor,
and,
can
yes,
you
go ahead.
repeat that? What did you just say? You're going to set up a...
I'm going to post a short problem set, and it's going to take you through things like the replication, transcription, and then translation. And the problems actually illustrate the processes. So the problems will actually help you to solidify your understanding. And,
okay, and then I saw there was another interactive exercise. When is that due? You posted
oh, Lord,
it.
I think I said that's the Wednesday of the Thanksgiving holiday.
The next one, not the photosynthesis, the new one, right?
Yes, yeah, the new one, which is it's the DNA worksheet.
Okay, and you said when is the due date?
I think it's the Wednesday of Thanksgiving.
All right, and then I also have a question about last week, Monday, there was no recording, right?
Yes.
Oh,
yes.
Can you send it again? We didn't, yeah, I didn't get anything. Can you send it again?
Oh, no, that's really important. I got my copy. Please, won't you just check? Sometimes strange things do happen. I will check and I will send it again. It's very important.
That was the 18th, right? Yeah.
Correct. Yes, the Monday. No, I did definitely. I've never missed one.
Let me just.
I'll check and see. Thank you for telling me.
Yeah, I don't have it. I don't have it in my inbox.
All right.
Maybe it was because it was part of the computer crash you were talking about.
Oh Lord, I hope not.
But you can just send it again. It'll be fine.
I'll send it. I'll just send it again, just to be sure. Yeah, I will definitely do that.
And then can you also? I sent you an email about some of the exam questions. Did you get a chance to look over them?
I haven't yet, no, but I will do so.
Okay, thank you so much.
I'll make sure I do. All right then. Okay. Anyone else? Okay, everyone. And I'll see you then on Monday, next Monday. And I'll make sure that the last Monday has been reposted to everyone. Very important one. Okay.
Sorry, one more question.
Go ahead.
Did you post the submission for the photosynthesis extra credit by chance?
Yeah. Yes. That's been up for a long time.
I can't submit it. It says like no submission or no due date or anything for some reason on Canvas.
No, really?
Yeah.
No. Oh, no. Just send it to me as an email. Just scan it.
Oh,
Scan
okay.
it and send it to me.
I
Yep,
did
that's
that.
fine.
Okay. Okay.
All right then.
Thank
Okay
you.
then. Okay. Goodbye, everyone.